All right, thanks. Thanks, Eric. And let's see. All right, so I have Kleefstra Syndrome gene therapy with a question mark because we're kind of at the very early stages of this, and it's really just trying to develop strategies, test them, at an early stage, and just really get started on this and try to figure out the feasibility. I've had a relationship with IDefine in the past, and at that point I said, don't work on gene therapy right now. The delivery tools weren't appropriate for this. There could be a lot of problems, and I think it's been kind of circling back here recently where there's been some really important technology leaps in the field that maybe the time is right to start to look at this. So I'm not going to talk a whole lot actually about what we're doing with EHMT1, except just kind of at a conceptual level, but I wanted to, yeah. All right. So I do a lot of gene therapy drug development in the lab, and I do have related to that. One would be Taysha, because that treatment for Rett came out of our group. So introducing here, AAV is the gene therapy vector that we're talking about, just to make sure that everybody's on the same page. This is a non-pathogenic virus in the parvovirus family. It's pretty ubiquitous in humans and other animals. And there's about 60 years of research into this virus since it was discovered that has kind of led to recombinant AAV that's been engineered as a gene delivery tool and a therapeutic. There's hundreds of clinical trials happening now using recombinant AAV going back to about 2000. Since 2012, there's been an increasing number of gene therapy products that have received full drug approval at the EMA and FDA. And basically, the virus itself is just this protein shell that's actually quite stable. You can keep it at -80 for at least 10 years if it's formulated right without any loss of stability. We've kept it in the refrigerator for a year without any loss of potency. And you basically gut out all of the viral genes and replace it with some DNA that you want it to carry. And you can engineer this virus shell. There's hundreds of different variants that have come out of nature, and now there's countless sort of lab-engineered variants of AAV that will target to different tissues. All right. So in terms of delivering a gene to the brain, there's three major methods. The traditional one that we use in the first clinical trial is if you wanted to deliver a gene to the brain, you stuck a needle in the brain and you injected the virus directly into the brain. This has been used. There's one approved gene therapy product for efficiency that uses this for any kind of approaches trying to get more widespread global delivery of a gene to the brain. This isn't very effective. So around 2009, 2010, there was a huge advancement where there's a version of AAV called AAV9 that was found that you could inject it IV, and a small amount of it will get across the blood-brain barrier. And that led to the gene therapy product for spinal muscular atrophy, Zolgensma, that was approved in 2019. Absolute phenomenal treatment. And then you could inject the same vector into the spinal fluid to concentrate the virus more around the nervous system. And then that actually, I think about a year ago, there was a version of the SMA treatment that was approved for intrathecal administration called Itvisma. So just my lab has been fairly prolific in terms of harnessing, really all these were with AAV9. We did the first intrathecal gene therapy trial at the NIH for giant axonal neuropathy, and since then have advanced this for multiple other diseases that have moved into cells. Rett here is on the list. And then we have other things that we're working on I'd say IND enabling, where we've talked to the FDA. We're in the midst of doing toxicology studies, drug manufacture, moving them towards trials. Multiple sulfatase deficiency just had their IND approved in June, and we're set to submit an IND for AGU in September, and Krabbe hopefully by the end of the year. And then we have Kleefstra Syndrome that just sort of jumped on this list that we're starting to work on. One of my tomorrow will be a lower expectation. Sorry, I think my slide shifted a little bit, but that's a circle. All right, coming to Kleefstra. I think that there's two, the general design of this, like I said, all of those trials that we've started, all the things we've been working on have been with AAV9. AAV9 is not good enough for this. It's not gonna be effective. So one of the things is using an advanced version of AAV9, and I'll talk about that a little bit. And then it's also we're coming into this with the idea that EHMT1 is going to be dose sensitive. We don't want to express too much. If we do, we can do harm. And so we can't use some kind of a generic expression cassette. We really need to engineer some kind of a regulator. So the idea here is that if we deliver this vector DNA into an environment with low EHMT1, like the patients, then we would get high activity of the gene therapy. And if we put it in an environment with normal EHMT1, then we would have low activity of the gene therapy. And this would also be an environment where, let's say we've delivered enough of the gene and we've treated the cell, but then there's more copies of if we get 10, 20, 50 copies of the gene into a cell, then it's going to have some kind of a feedback regulation. Okay, so those are the two kind of components that we're working on. One is the regulator, how to regulate properly, and then using this sort of advanced AAV. And the advanced AAV, we've got some variants of AAV9 that we've developed in the lab that look better than AAV9. We go with the best technology, and this actually came out of the Broad from Ben Deverman's lab. And this was published a couple years ago now about this AAV capsid that was reprogrammed to bind the human transferrin receptor. So this is a receptor that naturally moves macromolecules across the blood-brain barrier, specifically transferrin. And this is just I'd say the main exciting figure from the paper, where they put a small 7-mer peptide into the AAV capsid that they selected to target that receptor. So this is AAV9 injected IV, encoding a gene for M. scarlet. And these are mice that were humanized to express the human transferrin receptor. So AAV9 at this dose, you don't really see anything, but then their variant that they made, you see the whole brain basically lighting up. I think that they were estimating that it was at least 50 or 60-fold more potent than AAV9 to cross the blood-brain barrier. It's very specific for the human receptor, so if they use modified mice, the capsid doesn't work. And it actually doesn't work in monkeys either, or rats or dogs or any. It's very specific for the human receptor. Now, this is the first generation. They've made a generation of this called CapX. And in our hands, we've estimated that it's about 300 times better than AAV9. So this is just a snapshot of some of the experiments we've done in our lab, kind of preliminary work with CapX. These are 6-week-old mice that are injected and then sacrificed at 4 weeks post-injection. And then this is RNA-scope, a little bit washed out, but this is RNA-scope where all the red transgene expression. And I want to emphasize that Zolgensma, the FDA-approved product for spinal muscular atrophy, it uses a dose of, I think it's 1.2 times 10 to the 14 vector genomes per kilogram. So this is over 200 times lower dose than Zolgensma. And the amount of transduction here is actually a lot higher than you would get if you were using AAV9. All right, and then this is just zoomed in here at the cortex. It really go anywhere in the brain, it's going to look like this. And at that low dose, you don't see anything really with AAV9. And then CapEx, we see very extensive. I mean, by eye, it looked like about half the cells are transduced. So this is really a game changer for the whole field of gene therapy. Everything that we have that's not very close to the clinic, we're moving over to this technology. And the two with this right now are, one, you can't use it in a wild type mouse. You have to back onto this strain that has the humanized transferrin receptor. So that adds about 6 months of breeding time before you have a mouse model that you can test this in. The other thing is that this has not been in humans yet, but there's a few programs that are very close. One is a gene called Frizzle. That the cargo was developed by one of my colleagues at UT Southwestern. There's a foundation moving it. They say that they're going to be in humans by the end of this year. Okay. But I will say that the receptor itself, that's being used for a wide spectrum of biologics that are trying to target the brain, and that class of drugs hasn't really run into any complications in terms of disrupting the blood-brain barrier, damaging the blood-brain barrier, et cetera. So I don't know, we're all hopeful. All right, so that's the delivery side. I think that's what we're counting on, is that this CapEx is going to do what we need to do. The question is how to engineer the transgene cargo. And so we're kind of in this Goldilocks situation where we have to provide enough gene expression to provide a benefit. We need to avoid overexpression of the transgene. And so ideally, we would engineer a transgene cassette that responds to the cell environment to self-regulate itself, just like Goldilocks and the Three Bears. Not too little, not too much, just right. So if you're going to approach that as a problem, then this would be our basic AAV transgene, vector genome design. We have a promoter, gene, poly A. You could try to do a cell-specific promoter, endogenous promoter, regulate sort of how this would be expressed differently across different cell types, but it really doesn't control for transgene copy number most of the time. So if you get 1 copy, you're going to get 1x. If you get 10 copies of the gene in the cell, then you're going to get 10x. It's not necessarily really. You can have drug-activated promoters. I'm not aware of one of these that's in the clinic, but there's preclinical examples of this, where you have a promoter that's off, and then you add some exogenous drug, and you turn the promoter. One that requires this exogenous drug or compound, to activate the promoter. It also doesn't. So if you've got some cells that have 10 copies and some cells that have 1 copy, then you're going to raise and lower all of those, turn them on and off like a light switch. There's approaches where you can have the protein, where you engineer this to have some kind of a destabilization domain, and then you add an exogenous drug to stabilize the protein. Again, this doesn't really control for copy number. It's more like an on-off switch. And you can have tissue-specific microRNA binding sites. And this is a really nice way, say if you know that you deliver the gene everywhere, but it's more in a specific tissue like the liver, then you could engineer a binding site for a microRNA that's only expressed in the liver. And that way it's kind of a way to let it express everywhere, but just turn it off in one place. This is nice because these microRNA binding sites are really small. So they're economical in terms of the limited DNA capacity of AAV. Okay, so coming back to Kleefstra, kind of our approach here is we have, the ITRs, these are the AAV packaging elements. We have a limit that's really capped out at almost 4.5 kb. And we've got the EHMT1 coding sequence that's about 3.9 kb. So that only leaves about 550 nucleotides to fit the promoter, the poly-A, and all the system we need to engineer. This is a big ask. And I'd say that we're going down this. We have some preliminary data that for kind of a first generation shot at this, where we put our construct in cells that were EHMT down and we saw upregulation. But it's early. Give us time. We're working on it. So I am going to talk about Rett syndrome just to kind of give an example of where in the past we've faced this same problem and we've had a measure of success. And if you're not familiar with Rett syndrome, there's a lot of parallels here. In this case, it's an X-linked gene. MECP2 is a broad chromatin binding factor. It affects the expression of hundreds of other genes. There's lots of things that go wrong with the cells when they lack MECP2. Because it's X-linked, it affects almost all girls. It's typically fatal before birth in boys. And in this case, like half the girls never walk, half the girls never talk. Median lifespan about 40 years. But there's also something called MECP2 duplication syndrome that's well documented, that if patients are born with a duplication of this gene, they have a disease essentially just as bad as Rett syndrome. So this is your classic kind of Goldilocks gene. 2 times too much is bad. Not having the Okay, so this is kind of a nightmare from a gene delivery standpoint, because you have this situation where, because of random X inactivation, all the girls are MECP2 expression. Half their cells have functional MECP2, the good copy, and then half the cells have the good copy silenced, so they have dysfunctional MECP2. So you have this mosaic pattern. So if we have this hypothetical perfect gene delivery of MECP2, then we'd end up with a situation where all of our functional cells are treated, and then all of our previously normal cells are now dysfunctional and have duplication syndrome. So, trying to have some kind of broad, non-specific regulation for this wasn't going to work. So the solution to that was this design. We did incorporate the MECP2 promoter. We actually engineered a miniature, or we used a miniature MECP2 that was engineered by colleagues of ours in Scotland. And then the secret sauce was this element that we called mi-rare, which was this microRNA responsive element. And this was identified kind of in a combination of bioinformatic approaches and empirical approaches, where we basically found that there were a number of microRNAs that whenever MECP2 is up, these microRNAs are up. Whenever MECP2 is down, these microRNAs in the cell are down. So we selected a panel of those microRNAs, built-in binding sites for, and then this is kind of illustrated here, where we have these microRNA targets in mi-rare that we engineered. The vector doesn't express any microRNAs. It just has binding sites for cellular microRNAs. So this goes into a cell. We express the message. That expresses MECP2. If MECP2 levels go up, then it's going to drive those cellular microRNAs up, and then they will come back and they will bind to and downregulate the transgene. Then these microRNAs are already elevated, and they just silence the transgene right off the bat. All right. So, again, just to illustrate this, if we have MECP2 expression without any kind of feedback loop, then this is basically here we have transgene expression on the y-axis. We have increasing copies of MECP2 per cell on the x-axis. This would be MECP2 null cells, where we want low levels of expression that are going to be therapeutic, but then as we increase copy numbers, it's going to cross this threshold where we get overexpression. So in MECP2 minus cells, then we have this dose response where it can be therapeutic or it could be toxic. Into a wild type cell is just toxic right off the bat. If we have the regulated, then if we go into a MECP2 null cell, it's going to permit expression, and then at some point it's going to level out and plateau. And then if we go into a wild type cell, it's just going to be repressed from the beginning. Okay. So we did experiments of this in mice where we used a MIC-tagged MECP2. Without this regulatory element, we could readily visualize expression of MECP2 transgene after dosing wild type mice. These are in wild type mice. Then with this element, we suppressed the expression. It was still there. If we the expression was still there, it was just highly knocked down. And then this is just where you kind of zoom in, where without the regulation, we got similar expression in knockout mice or wild type mice. With this regulatory element, we blocked expression in wild type mice, but it permitted expression in knockout mice. Okay, and just to illustrate the impact of this is a survival curve in wild type mice. This is a safety study. The blue and the green lines are different doses, high doses of our unregulated vector that lethality in wild type mice, and then our regulated vectors were tolerated well. And we also did the same thing looking at this Rett behavior score. Higher on this y-axis means more Rett-like behaviors. So at the high doses of the unregulated vector, we induced Rett-like behavior. But our regulated vector, the high dose, that tracked along similar to vehicle injections. Again, these are just wild-type mice. This is a safety study. But then there was a pretty comprehensive efficacy study that was published a couple years ago, treating mice at birth, 7 days old, 14 days old, 28 days old. There were improvements in survival, weight, severe breathing abnormalities, gait, hind limb clasping, this aggregate Rett behavior score. The degree of rescue was dose-dependent and age-dependent. Okay, and then, of course, the next part of this story is this technology was all licensed to Taysha Gene Therapies, a startup. They moved this into a clinical trial where they dosed the first patient in 2023. They're now dosing their phase 3 study. And this is from a year ago, but this was a poster that they presented, or a figure from a poster that they presented that kind of summarized their phase 1 data. This was 10 patients treated with this gene therapy treatment for Rett. They aged from, I think, 6 years old to early adults. And so all 10 patients saw improvements, including the adults. And there were things of, instances of patients being nonverbal and then using phrases with meaning, no purposeful hand use, playing with toys, transferring them intentionally, non-ambulatory, walking with support, requiring a G tube for feeding to go into oral feeding. Just an example. They had, I think all of the assessments were videotaped. They went through independent reviewers that evaluated these, and came to consensus scores about improvement. And I should say that at 6 years older with Rett, just basically natural history studies over decades with over 1,000 patients, I think there's something like less than a 1% chance that they would ever gain these skills after 6 years old. So this was very compelling, and they're enrolling. They might have finished enrolling their phase 3 study, and this is going towards kind of a rapid path of drug approval. So coming back, we've got some other strategies besides the microRNA regulation that we've been doing for things like Fragile X syndrome, DDX3X, MEF2C, FOXG1. Actually, we're doing some work on that to try to. So we're working on this. Hopefully next time we meet, hopefully long before then, I'll have a more complete story here. But our next steps, we've been working to investigate avenues for sort of engineering a de novo feedback regulation system for EHMT. We need to package this in the CAPEX AAV capsid, and then test the regulated constructs to see if we learn some match cells, evaluate safety in wild-type mice, and then kind of working to set up. We've collaborated with Jackson Laboratories on a number of studies throughout the year, and I think they're working on getting the EHMT1 mouse models up and running, cross to the transferrin receptor capsid or transferrin receptor background, ready if we have a lead candidate that they could test it there. So with that, I'll thank my lab members, especially Hiri, who's right here. She's the one who's leading this work. She's been in my lab for about 8 years, does great work, and sources of funding, and IDefine is funding all of our work on this. So thank you.